Hello, everyone, and welcome. My name is Elise Malavasi, and I am the Associate Director for Neuroscience at Concept Life Sciences. It's a real pleasure to open today's webinar entitled Uniting Biology and Digital Drug Discovery to Identify New Therapeutic Opportunities in Neurodegeneration. We're delighted to have such a global audience with us for this important discussion. As you know, neurodegenerative diseases remain one of the greatest challenges in today's healthcare. But by combining biological insight with powerful digital technologies, we're now opening exciting new opportunities for discovery and treatment. Today's session will show how these approaches are coming together to accelerate innovation. We are joined by two speakers, myself from Concept Life Sciences, and I'll be sharing perspectives on the biological foundations of this work and the recent experimental advances, and Dr. Samuel Walters from Delta4 AI, who will walk us through how digital platforms and AI-driven discovery are reshaping this field. We'll wrap up with a Q&A session. So please do submit your questions in the chat anytime during the presentations. And with that, let's get started. So today I will take you through some of the work we have been doing to characterize and validate the use of iPSC-derived astrocytes as a tool for drug discovery and development in the field of neurodegeneration and neuroinflammation. So why did we choose to focus on astrocytes? As a cell type, astrocytes have been quite broadly neglected by drug developers, being regarded as little more than ancillary cells in the central nervous system. However, recent discoveries are overturning this perspective or perception by demonstrating that astrocytes, in fact, play a very active and prominent role in neuroinflammation and neurodegeneration. So astrocytes are the main glial cell type in the central nervous system, and under physiological conditions, so in a healthy brain, they are a very nurturing, supporting, caring cell type, without which neurons simply couldn't function. However, in the context of neurodegenerative diseases, astrocytes downregulate all their neuroprotective functions and acquire pro-inflammatory and neurotoxic phenotype that then further exacerbates damage. So in this pathological context, activated microglia secrete a plethora of pro-inflammatory factors, and it was demonstrated in seminal papers that a combination of three of these factors, TNF-alpha, interleukin-1 alpha, and C1q, in combination are sufficient to drive the phenotypic transition of astrocytes from a neuroprotective to a reactive neurotoxic phenotype. And once polarized, astrocytes start secreting their own cytokines and chemokines. And in addition to that, they also release molecules that are directly neurotoxic to both neurons and oligodendrocytes. So our deepening understanding of the molecular pathways that are involved in this phenotypic transition of astrocytes from neuroprotective to neurotoxic really creates the opportunity to approach the development of drugs targeting neuroinflammation from a new angle. And in addition to that, since both astrocytes and microglia become reactive early on during the course of a disease, this also offers the opportunity to broaden the time window for therapeutic intervention. However, these opportunities also come with some challenges. If we want to test drugs that can counteract this phenotypic transition, we need to have at our disposal robust and reproducible assays that are based on accurate and translational and accessible in vitro models of the neurotoxic reactive phenotype. We also need speed and scalability to support the design, make, test, and analyze cycles that are at the core of the drug discovery process. And so, faced with these challenges, we decided to evaluate pre-differentiated and cryopreserved iPSC-derived astrocytes. These cells have several features that render them attractive to us. First of all, they are a human model, and therefore they offer translational value. They are serum-free, which is really important because we know that serum induces a phenotype that is already reactive in astrocytes, so we don't want to have any serum in the medium. They are differentiated in a consistent and standardized way. They offer minimal turnaround time, so that element of speed that we really need. They have a consistent genetic background, which is useful and desirable at the earlier stages of drug discovery, when we need to directly compare many compounds, and finally they have consistent and assured quality. So our objectives were, first of all, to determine whether these iPSC-derived astrocytes can reliably model the neurotoxic reactive phenotype that we're interested in. And secondly, we want to determine whether if we were to polarize these cells over and over again across the seasons, and using different operators and different lots of cells, we could observe reproducible responses. And finally, we wanted to determine whether this model and assay can be useful not only to test and validate existing targets and drugs, but whether it can also help us predict completely new drug targets. So for these experiments, we plated cells in 96 well plates, and we allowed them to establish in vitro for 8 days. So that's to allow the cells to really stabilize and recover from cryopreservation. And after 8 days, the cells are then ready to be treated or assayed. So the aim of the first experiment was to transcriptionally profile the responses of these cells to a stimulus, a pro-inflammatory stimulus with the so-called TIC cocktail, which, as a reminder, is TNF-alpha, interleukin-1 alpha, and C1q. So the cells were stimulated for 24 hours with this or with vehicle. And then at the end of the stimulation, we extracted RNA and performed RNA sequencing. And we also collected cell culture medium to perform profiling of secreted cytokines and chemokines. And in the second experiment, we aim to validate a positive control that could prevent the polarization of these astrocytes to a neurotoxic reactive phenotype in response to TIC. So the approach was very similar. Again, we allowed the cells to to establish and stabilize for 8 days. But then in this case, the cells were pre-treated with dexamethasone or vehicle for 1 hour before receiving the 24-hour TIC stimulus. And then after the stimulus, the cells were lysed, and we performed RT-qPCR to quantify the changes in the expression levels of several marker genes. And we also again performed cytokine secretion profiling. And then we used the results from both of these experiments, so the transcriptional profiling and the positive control validation, to ask how reproducible these responses were across different lots of cells. So in collaboration with our partners at FIOS Genomics, we first of all determined that at baseline conditions, iPSC-derived astrocytes express key differentiation markers, and they do so with very limited lot-to-lot variability. So you can see the circles here are data generated from one lot of cells, and the squares are data generated from the second lot of cells that we analyzed. And in most cases, the relative expression levels are very comparable across lots. These here are markers of mature astrocytes that we would expect to see expressed in these cells. So it's reassuring to see that that's the case. And at the whole transcriptome level, we see that when comparing again the two lots at resting conditions, we see that whilst there's a number of differentially expressed genes, neither the magnitude of these differences or the significance of these differences is particularly high. And we also observed that most of these genes are involved in cell cycle control rather than inflammatory responses. So in the next experiment, we again analyzed the responses to TIC stimulation, comparing again two different lots of cells. And so the volcano plot on the left shows that, first of all, there was a very large transcriptional response to TIC stimulation, as we would expect, with many upregulated and downregulated genes. In total, we observed just under 5,000 differentially expressed genes. And amongst these genes, reassuringly, we saw that several of the key markers of the neurotoxic reactive phenotype were strongly upregulated in both batches of cells. So as you will see, for some of these genes, the fold changes were different across batches, but the changes were always occurring in the same direction, which is what we would expect. Again, looking at the whole transcriptome, when we performed congruence analysis to compare the TIC responses in the two batches of cells, we saw that these were, as expected, significantly positively correlated with each other, with only a handful of genes changing in opposite directions in the two batches. So essentially, this confirmed that the cells are responding to TIC stimulation by upregulating the key markers of the neurotoxic astrocytes that we expect, and also they do so consistently across batches. Next, we wanted to ask whether the responses we observed in these astrocytes are consistent with what was reported by others and observed by others using comparable but slightly different models. So for this, we decided to compare our data set to the data set that was generated by Barbar and colleagues and published a few years ago. Here's the reference on here. And just as a summary here, I wanted to remind you of our methods and how it compares to the method described in the Barbar paper. So we started from cryopreserved, pre-differentiated human iPSC-derived astrocytes from a single donor. And they came from 2 different lots, so 2 different differentiations. They were allowed to stabilize in vitro for 8 days, after which they were stimulated with TIC or just vehicle for 24 hours. And then we extracted RNA to perform sequencing. In the Barbar study, by contrast, they started from iPSC lines from 3 donors of different genders. They established iPSC colonies and then differentiate up to mixed neural cultures. These mixed neural cultures were then subjected to purification to isolate a purified astrocyte population. And then these astrocytes were either left unstimulated or stimulated with TIC for 24 hours before performing the RNA sequencing experiment. So according to the authors, the entire process from iPSC cells to purified astrocytes can take up to 10 weeks. And that's in contrast to the 8 days that were necessary using our method. So there was a significant time saving. And interestingly, when we compare the transcriptional responses in the 2 cell populations, so the CLS data set and the Barbar data set, we again observed significant positive correlation between the 2 in the way they responded to TIC. And again, there was only a handful of genes that changed in opposite direction. And we're looking at functional pathways, so doing pathway analysis in the 2 data sets. Again, we observed that there was a very good agreement between the 2, with pathways such as signaling by interleukins and interference signaling being strongly upregulated in both data sets. And we also observed that eukaryotic translational elongation pathway was downregulated in both. So that gave us confidence that the iPSC-derived astrocytes that we've chosen to utilize as a model are appropriate to really model this neurotoxic reactive state. And next, we wanted to validate a positive control that can predictably, reliably, reproducibly prevent the transition of astrocytes to this neurotoxic state. So we chose to use dexamethasone for this, and we tested the effect of dexamethasone in 3 different batches of astrocytes. As expected, we saw in response to TIC stimulation a strong upregulation of the expression of pro-inflammatory genes like interleukin-6 and markers of the neurotoxic reactive phenotype like GBP2. And reassuringly, we observed that these gene expression changes could be reproducibly prevented by pretreatment with dexamethasone across lots. And we also looked at the cytokine secretion profile in 3 batches of cells, and as expected, we saw that several cytokines and chemokines were strongly regulated in response to TIC, and this upregulation was counteracted by pre-treatment with the TIC, with dexamethasone. And really, to further reassure ourselves that the dexamethasone responses were reproducible across lots, we took a closer look at the absolute levels of secreted interleukin-6 as an example of a pro-inflammatory marker. And so across lots, we did see the expected increase in secreted levels in response to TIC. And we saw that reduction when the cells were pretreated with dexamethasone. But we also observed that different lots of cells respond with different strength to the TIC stimulation. Some secreting more and some secreting less interleukin-6. However, when we looked at normalized data, so removing that element of variability between absolute levels of secreted interleukin-6, we see that across lots there's around a 30% residual secretion after dexamethasone, which means about a 70% reduction in the responses. So that confirmed that, as we were hoping, the pro-inflammatory responses across lots can be reproducibly downregulated by dexamethasone. This final data essentially confirmed that we validated a neurotoxic reactive astrocyte polarization assay with an appropriate positive control. And so our next objective was to really leverage our model and assay to determine whether we could use this information to help us identify and validate new drug targets. And this is where I will hand over the stage to Samuel, who will take you through the next part of this presentation.
Thank you very much, Elise. Also, a warm welcome from my side. I'm Samuel from Delta4, and will guide you through that part 2. And also have to emphasize that everything which you just heard is the basis of what we in digital drug discovery require to model properly, and is exactly what we need to create great models and create new insights. Again, a warm welcome, and just to give you a short introduction where I come from with Delta4. We are a Vienna-based small tech bio company with our prime goal to deliver new insights to pharma companies in the context of early and late stage drug development with drug combinations, indication expansions, target and biomarker identifications with a broad range of areas we've already got experience and achievements in, from nephrology to hepatology, oncology, as well as rare diseases, and today as well with neurodegenerative diseases. And in this context, just to give you a small overview, there we are, where today's project is embedded in. So we have parts of our work in early stage drug discovery, from target identification and target prioritization, as well as disease-centric repositioning, which is today's topic, which then further leads to preclinical model assessment, and from that onwards can go to biomarkers, side effect modeling, drug combinations, and indications expansions as well. But today's topic is primarily early stage drug discovery with the basis which was already laid before. And to give you roughly an idea what data and what information we use to achieve all of those insights, we utilize a vast area of biomedical data, which we combine with solid AI architecture for data analysis. And with our iPSC framework, We utilize core data sets from PubMed, clinical trials, and patents for already established knowledge and combine them with dedicated biological databases with Camber, Mesh, and Ensembler for drugs and phenotypes and genes, for example, with additional multitude of databases for pathway information, mechanistic insights, and preclinical data, and so forth. And combine these with omics data sources, and particularly very important, as also with today, with customer or project specific data, which is already created for this particular project or model of question. And with all of these data and databases, we combine them in an organized structure with knowledge graphs, relational databases, and all of our curated processing methods, which try to not only take the data as it is, but also elevate, curate it to get rid of false positive signals and to really drive novel insights and novel ideas. And to give you a bit the whole context for today's question, we have here, again, to reverse the pro-inflammatory phenotype in human iPSC-derived reactive astrocytes. So for that purpose, we utilize all of the data which has already been created and subsumize them with ontologies, literature data, the omics data created by Concept Life Sciences, as well as all of our catalogs, and combine them in an astrocyte model where we also created two subsets of that model for the neurotoxic and neuroprotective phenotypes. And we utilize our model of reactive astrocytes, screen them against our drug library. With network interferences, we look at mechanisms and other evaluations, then to distill novel drug prospects, of where we will have a closer look further on. And I will guide you now for this process in detail. The very first step and the most critical step here is actually to construct the astrocyte model from all of the knowledge which is available. And based on the catalogs we have in our iPSC framework, we utilize literature ontologies such as gene tissue associations, gene mechanism associations, gene tissue for astrocytes, gene mechanisms for neuroinflammation, and relevant mechanisms. And by that, we can already distill 275 genes in this context. We expand that with the RNA-seq data analysis and all of the differently expressed genes from the stimulated iPSC astrocytes, which lead to another 156 genes. We expand that further with structured data such as the Human Protein Atlas, where we can look which genes are astrocyte specific, which do play a critical role not only in the data, but might be interesting to look at as well, which gives us a further 51 genes. And then to have a detailed look at our question here for the neurotoxic and the neuroprotective phenotypes, we go to selected core literature. Terms such as neuroprotection sometimes are not available in mechanisms catalogs. And then we can directly go to the source and see what has already been published in this area. And by this way, we ended up with another 97 genes. Some of these parts overlap. And in total, we received a model of 522 genes with the respective subsets for the neurotoxic and the neuroprotective phenotype of 37 and 39 genes respectively. And you can see here this visualization will keep on where the orange badges represent the neurotoxic and the dark blue patches represent the neuroprotective subset of the complete astrocyte model. And with this astrocyte model, we can now evaluate what kind of mechanisms, what kind of pathways can we see which play a role in how we modeled these reactive astrocytes. And we do this by looking at the classic mechanism enrichment, but we further expand the network, the model we have, and we transfer it to our iPSC protein-protein interaction network. And in this protein-protein interaction network, we have embedded all of the mechanisms, various phenotypes, various drugs and their interferences, as well as information such as genuine drug targets for respective drugs, or embedded. And also in this way, we can embed information we already have from omics experiments to evaluate which associations are of higher and of lower importance. So we can change the weights of all of these gene to mechanisms, gene to phenotype, gene to drug information from already all of the previous knowledges, as well as with these omics data sets we can integrate here. And by that, we can obtain a mechanism network centrality rank, kind of giving us an idea, not only that there is a pathway or a mechanism which is enriched in our context, but also particularly to the right side, you can see it here, for our neuroprotective and our neurotoxicity subset. And by that, we can, for example, see signals such as for VEGF signaling. We can see that in neuroprotection, this actually has a higher centrality score. And if we go back to the literature, we actually can also see there is already information that VEGF signaling actually plays a critical role in neuroprotective mechanisms and how the cell is able to a certain dose. And with the model and all of these mechanisms, we can now screen against our drug library of over 9,000 drugs. And we will have a closer look at one of our top candidates here, which is istradefylline. And in the center, you again see our astrocyte model with the neurotoxic and the neuroprotective subset in orange and in dark blue. And to the right, you see the istradefylline model with light blue badges attached. And there is a substantial part of the istradefylline drug model, which interferes with our astrocyte model, partially also with the neuroprotective set. And of most interest is the adenosine A2A receptor, which we already can also confirm from literature is playing a critical role and is also upregulated in our omics dataset, whereas istradefylline is a selective antagonist of said receptor, therefore counteracting actually the neurotoxic behavior we see. So this is a very promising target to counteract our neurotoxic phenotype to get back to our neuroprotective state, which we want to do. And to give you a bit of a more idea of how we can look at these models, you see here the model again in our interactive model viewer. We can see the complete model here again highlighted with also the interfered subset of where the istradefylline model is interfering. And again here now highlighted the istradefylline model as a whole, where the small right part is not part of the astrocyte model and all of the left parts are the astrocyte model itself. We can evaluate where all of the information is coming from. We can highlight which of all of our nodes are actually based on our omics data. And see where or which are based on from other sources, and to see where this information is actually originating from. With all of our omics data, we can now highlight also which of all of our nodes are actually positively upregulated in this case. We can have a further look of all of our nodes in our network, which of them are a drug target of any genuine drug, which are now highlighted and also moved to a star for visualization, particularly also our adenosine A2A receptor. And utilizing all of the information at our hands from various sources, we can investigate what to do with this model. And not only can we look at drugs, we can also have a look at our explored mechanisms of neurotoxicity, neuroprotection, in which phenotypes do they actually play a critical role. So we had a look at our complete phenotype library of over 9,000 phenotypes, but here I particularly focus on nervous system diseases. And you can see here on the left side a sunburst where already the accumulated interference scores show that our neurotoxic and neuroinflammation plays a stronger role in central nervous system diseases, but for example, a reduced role in relation to these in muscular neurodegenerative diseases. And if we combine the network as we have before with all of our mechanisms, we can again see here now to the right side, we have astrocyte activation, differentiation, and various mechanisms. And that particularly, for example, for astrocyte activation in Alzheimer's disease, all of these diseases have high interference scores with our model. We see that astrocyte activation has a relative higher centrality score. And if we go back to literature, we again can confirm that there is evidence that in, for example, large-scale proteomic analysis, there have been metabolic changes which were shown to be associated with astrocyte activation in astrocytes particularly. And so by combining our model with our phenotype library, together with all of these mechanisms and pathways we have available at our hands, we can have an in-depth look at what actually really happens when we investigate the modeling compared also with the information of our omics dataset. And from this step onwards, Where can we go to? Is the slide moving? Yes, there we are. Where can we move on from this? So we have already shown now how to identify and target with our model a drug target and a mechanistic evaluation of one drug in one disease. We can look for further targets. We can look for further diseases in depth. We can move on for bioinformatics identification, side effect modeling, and drug combination analysis. And all in all, lay the foundation for further preclinical experiments to, in the end, have a novel insight and really come up with new therapeutic avenues to cure diseases in the long run. So to sum up today's talk, by Elise, we already could demonstrate that by combining our experimental biology and digital drug discovery expertise, we could demonstrate how iPSC-derived astrocytes can be successfully applied to model the neurotoxic reactive phenotype. We could show how a reproducible tick-induced polarization assay was also validated with pro-inflammatory markers, and all of this information could be transferred into an in silico representation for mechanistic evaluation and also identify drug targets, candidates, the context in various phenotypes, and by that lay the foundation for further experiments and knowledge creation. And with this, I want to thank you very much for your attention, and we are open for any questions you might have.
I can see there are some questions in the chat. So the first one is, what was the difference between our study and the Barbar study, and was your study superior to the previous study? I wouldn't comment on superiority. I don't think that was really the point here, but the main difference that we were trying to examine by comparing these two data sets is starting, so in the Barbar study, they started from iPSCs, and they allow them to differentiate and into mixed neural cultures, and then the astrocytes were purified from these cultures. So all this was happened, without any freezing steps in between, and that is why it took several weeks to go from iPSC to assay-ready astrocytes. With the model we use, because they are cryopreserved after differentiation, we were ready to assay them after a day. So really what we wanted to ask is, does this time gain come at a price? So are we losing biological representativeness in this model? And this is why we wanted to compare it to other similar models, and the answer was that actually you can cryopreserve these cells after differentiation, bring them back and get your results faster, and the results will essentially reproduce what you would see if you started from iPSCs and waited for 10 weeks or longer. And it was a question about whether this model can work in solid CNS tumors. So we know that astrocyte reactivity can contribute to tumor growth, can create a supportive microenvironment in CNS tumors. However, this the one we describe here is purely an in vitro model. So we haven't really explored any potential interferences with tumor cells, but it's an interesting question. Question. I'll just want to see if there are any questions for specifically for Samuel.
There was one on me on what ontologies we base our work on.
Yeah.
And that is, we don't have, we integrate many ontologies. So we have our RPC framework where we integrate public available information and enrich that with our own data and cross-reference it with what we have available. So it's not one singular ontology publicly known, but there is ontologies such as Go, Reactome, and all of these data integrated into our knowledge graph and iPSC framework, where we add on top of it and modify it for our custom use for these models to have it precisely as we wanted to do. All of the public ontologies always have some small quirks which might not 100% fit the project in need, and all of that work is done in-house to have it at the best use for whatever modeling and analysis we want to do. And thank you very much.
So, do DEX-treated human astrocytes also secrete interleukin-1 beta? So we haven't assessed interleukin-1 beta secretion in response to dexamethasone, but we did do measurements of interleukin-1 beta in resting versus stimulated, TIC stimulated astrocytes. And what we saw was that the secreted levels were very, very low in both groups, and there was no difference in the levels after pre and after stimulation. And generally, astrocytes do not secrete large amounts of interleukin-1 beta. And what are the results in immunocompromised people? We can't answer that question, unfortunately, because we have not studied any responses in vivo. This is purely an in vitro model. So yeah, I can't answer that, I'm afraid.
There's another question. What indication the studies gives with respect to the translational output to multiple sclerosis, if there are any different types of MS, and which type was included in the study? We did not make a deep dive into MS yet, so this is a follow-up option we have. For the phenotype catalog, we have over 9,000 phenotypes included, which is multiple sclerosis and multiple subtypes primarily available in MESH. But as always, the moment we do a tailored analysis of a topic, we can add custom and information which is not available directly. But from the get-go, there are over 9,000 diseases, which includes multiple subtypes. I don't have them at the top of my head yet, but all major available ones are included in the interference screen for this analysis. And the same goes for the clinical scenario. This is so far preclinical analysis, so there is no clinical data in that particular aspect, at least from the model we have here yet. And there's some more questions for you, Elise, I believe.
You can see, can we test beneficial effects of extracts or plant extracts in with these models? They are completely modality agnostic. It's a very completely modality agnostic model. So whilst so far we have mostly focused on small molecules, I don't see why it wouldn't be suitable for testing biological extracts, for instance, including plant extracts. And can you give some insights on co-culture systems? That's a very interesting question and something we've given a lot of thought to. So it is known that astrocytes as well as microglia tend to respond a lot more strongly when they are cultured in isolation compared to when they are cultured with neurons and other glial cell types. So we do expect to see differences in a co-culture setting. and that is definitely something we want to investigate further. See, how does the magnitude of the astrocytic response changes when we co-culture them, say with neurons and microglia. I think we have run out of time for live questions, but if you do have any further questions, please keep submitting them in the chat, and we will collate all unanswered questions and respond via email.
Thank you very much for all the questions.
A recording of the session will be available soon, and we encourage you to look out for our upcoming webinars, where we'll continue exploring innovations at the intersection of science and technology. Thanks, everyone.